Hi everybody, this is Brenda and we are in MOT 503 Neuroscience for Occupation, week 1. And this week we're going to do Central Nervous System Overview Part 1. I was planning on doing this in 2 separate videos, but I think that we can do it in one, and I think it'll be a little more cohesive that way. I just thought it might be a little long. But if you do get tired, feel free to stop. I know watching a talking head in a slideshow is not nearly as much fun as being together in person. But the information is still really good. You're going to need to know it. So, take your time, get some popcorn, stop it as many times as you need to, and get through the information. And we will get through it together. So, for this first week, don't forget to do your syllabus quiz. It is due at the start of lab time on Friday, which is going to be 10 a.m. As I told you before, get together with a friend. Do it on your own, redo it later if you want. You can do it multiple times. Mostly I'm just wanting you to get looking at the syllabus. It's under tests and quizzes in ACE. All right, after that, what are we going to be doing today? We're going to be looking at these items this week and next week, and actually probably into week 3 as well. But today we're particularly looking at the spinal cord and the brainstem. We'll go all the way up through the pons. So kind of starting low, going high, and learning about the central nervous system. But our goal for today and next week is for you to start recognizing these structures on sight. We want to orient ourselves to where we are in the brainstem and in the spinal cord, so that when you look at a cross-section or when you look at a gross anatomy view of these items, you're going to be able to recognize where you are and what it is that you're looking at. And later on in the semester, we will be able to know more about what those structures do. Right now, we're just getting oriented to where we are. We're also going to be looking at the main arteries of the brain and their importance and their impact on function, especially in terms of things like strokes. Okay, but first, let's review just a little bit of gross anatomy. Hopefully in your undergrad anatomy class and in your graduate anatomy class, you had some instruction on the nervous system. So what is the most basic unit of the nervous system? Do you remember? You remember what that was? It would be the neuron, okay? And what is the part of the neuron that receives the information? The part that is the kind of the feelers of the neuron. That would be the dendrite. Okay, one more question for you. What part transmits the information onward? What's that thing that projects from the body of the neuron, the cell body, to send that information on to the next neuron? It would be the axon. Okay, so that's our most basic unit of the nervous system. This is what it looks like. So we have the soma, or the cell body, and we have these dendrites that are sticking out from the cell body. They're receiving the information. And then we have the axon that is sending that information on to the next neuron. So that's very important to be aware of. All right, so how does that information get transmitted down the axon? Is it chemical? Is it electrical? Well, it's electrical going down the axon. Now, when the information jumps from one neuron to the next, then we have chemicals that come into play as well. So what are the most important chemicals that people need in order to make this transmission from one neuron to the next? We have to have sodium, potassium, calcium and chloride. So what happens when your electrolytes get off? People become motorically slow. They become clumsy. So a couple years back, my dad became ill because he had low sodium. And one of the ways that we knew that something was off was that he became very clumsy. He was falling. Just kind of seemed like weakness, like his legs wouldn't support him. And it all came down to his sodium being off. So information is transmitted down the axon. How? Electrically. And it jumps from one neuron to the next. How? Chemically. Those are important things to remember. And it's those neurotransmitters that send that information from one neuron to the next. Do you know any disorders or diseases that are caused by neurotransmitter problems, like not having enough of a specific neurotransmitter? Well, Parkinson's disease is one. So is myasthenia gravis. Also, some of our mental health issues can stem from not having enough neurotransmitters available in the system. So that's something important as well. So now we remember a little bit of the basic unit, the axon, the neuron with the axon and the dendrites. What do the glial cells do in the nervous system? Well, the glial cells are the supporting cells for the central nervous system, and they do a bunch of different things. They clean things up, they provide signals, they use chemicals, they're also the scavengers and clean things up. So let's talk a little bit about these cells. The astrocytes release calcium as a signal, and they are also taking up extra potassium as scavengers. So they are helping keep the chemical balance around the cells appropriate for transmitting that information on. We also have oligodendrocytes. These are the myelin coating within the central nervous system. Insulation is really important. If you think about like bare wires, not only are they dangerous, but they transmit information slower than insulated wires. Same thing in the nervous system. If we have myelinated neurons, they're going to be much, much faster than unmyelinated neurons. So when there are diseases of these macroglial cells, when they go away, they cause things like multiple sclerosis and Guillain-Barré syndrome, other issues because these macroglial cells, the oligodendrocytes, are not myelinating, or the myelin cells, because we have also some peripheral ones. We'll talk about those in just a minute. They're not doing their job. They're not myelinating. So oligodendrocytes actually work to cover several adjacent neurons. They have these projections that reach out and grab onto a few different neurons and protect them. In the peripheral nerves, we have Schwann cells that are going to myelinate peripheral nerves. They usually wrap around just one neuron, but there's occasions where they wrap around two or more. But Schwann cells in particular are looking at, are in the peripheral nervous system and looking at myelinating usually just one neuron. Now, these are the macroglial cells. We also have some microglial cells. These are the scavengers, and they'll clean up the environment, the neuron environment. These are phagocytes, which act as the immune system. So let's go on and talk a little bit more about how the nervous system is organized. Okay. For this, you're going to look under lessons to find more find the information that's on the next slide. You have actually like a handout type of sheet that's on lessons to find that. And I believe it's under like additional resources for this lesson. But let's briefly talk through this, and then I want you to be sure to review it, because even though we're only briefly reviewing it, these are things I expect you to know as an underlying basis for what's going on in the nervous system. So we have our central nervous system. What does that consist of? Basically the brain stem, the spinal cord. Okay. Then we have the peripheral nervous system, and this includes spinal nerves, but it also includes cranial nerves. These are also considered peripheral nerves. They have two components. So we see the sensory or afferent division, or the motor or efferent division. Sensory, afferent, going at the central nervous system. Motor, efferent, exiting the nervous system. So that's one way that you can remember afferent versus efferent. Afferent at sensory, motor, efferent, exit. Now the sensory has two divisions. We have somatic, which goes to basically our skin, and also joints, things like that. But it is touch, pain, pressure, vibration, etc. You can read it there. And then we have some special senses that are also considered part of somatic sensory. Visceral sensory tells us what's going on in our gut. They tell us when we're full. They tell us when we're hungry. They tell us when our bladder is full. So they tell us these sorts of things. And they are stretch, pain, temperature, chemical changes, any kind of irritation in the viscera, nausea, hunger. Okay, that's the sensory system. Then we have the motor system. We have somatic motor, which is general, innervating all of our skeletal muscles. Then we have our visceral motor, which innervates our smooth muscle, things like cardiac, glands, lungs, things like that. This is our autonomic nervous system, and it includes the sympathetic division and the parasympathetic division. So sympathetic division is our fight or flight. Parasympathetic is our rest and digest, or I've also heard it called feed and breed. It will include genitalia, sexual function, as well as digestion, things like that. And sympathetic works kind of in opposite, or kind of a tug of war with the parasympathetic, to help us when we are more stressed in that fight or flight mode. We'll be talking about those more as the semester goes on as well. So let's remind ourselves about some of the directional words that we use when we talk about the body. You should have these from anatomy. Cephalo, caudal. So cephalic is our head, caudal is our feet. Proximal, distal. Proximal means closer to our trunk. Distal means more extended out toward our extremities. Coronal, sagittal, and transverse. So coronal section is one that would go this way through our head, like someone was chopping sideways through our head. Sagittal is right down in this plane, like kind of through our body from front to back. Mid-sagittal would be right in the center of our body or our head, kind of in the midline. And transverse is anything that goes this way, a horizontal section. So those are the sections. Dorsal, which is your back. Ventral, which is your front. Anterior, posterior, different words, same thing. Anterior front, posterior back. Medial, more toward the middle. Lateral, more toward the sides. Ipsilateral, same side. Contralateral, opposite side. Afferent is at the central nervous system. Efferent, exiting the central nervous system. There are a couple exceptions to that, which will way later in the semester we'll talk about that. But right now, the main thing is knowing afferent at the central nervous system, efferent exiting the central nervous system. All right, let's talk spinal cords. We're going to learn a little bit about where we are in the spinal cord. Let's get oriented. That would be what? Gray matter or white matter? That would be the white matter. Got lost in my slides. I'm losing the page here. There we go. White matter. Okay. Now we have some anatomical regions. If you look in chapters 1 and 13, you're going to get a lot of the information that you're seeing in these next slides. Not all these slides are from the book, however. So there are these we talked about the 4 anatomical regions of the central nervous system, which includes the brainstem and cerebellum, the cerebrum, the peripheral nervous system, and the spinal cord. Let me kind of say them more kind of out to in. Peripheral, spinal cord, brainstem and cerebellum, and the cerebrum. Okay. If you look at page 6, figure 1-4, you'll see those regions a little bit better. So what kind of a section are we looking at in the spinal cord here? This is a transverse or a crossways section. So we're looking at the white matter. What's included in the white matter? What is traveling in those white matter areas? Is it cell bodies or is it axons? It's axons. So we have axons of all the different neurons going through these areas. They're white because they're myelinated. Now the gray matter, that kind of H-shaped or butterfly-shaped thing in the middle, is the cell bodies. All right, so here's what it looks like in an actual picture, cross-section. Now, is that red section, is that dorsal or is it ventral? Think about it for a minute. Dorsal or ventral? Take a guess. It is dorsal. This area is called the dorsal funiculus or dorsal column. It's also called the fasciculus gracilis because everything in anatomy has to have 2 different names to it just to make it complicated. All right, so that's the dorsal column or the dorsal funiculus, also called the fasciculus gracilis at this point in the spinal cord. Now, we have gray. What is this? This would be the lateral column. All right, pretty straightforward, right? And now we have yellow. Is that dorsal or ventral? It is ventral, which is also called what? What's the other word that we use for the front? It's anterior, so it's ventral or anterior. So this is the anterior funiculus or the anterior column. Are these white or gray matter? These are white matter. So what's traveling through these areas? It would be myelinated axons, what we just said just a minute ago. Okay, so here's a diagram of it. We're getting a little bit more oriented. So we have some names for the gray matter as well as the white matter. Okay, so in the gray matter, we have the posterior gray horn. We have the anterior gray horn. So you want to locate where those are. Now, what do you suppose is happening in these areas? That's where the cell bodies are. So we talked about how the outside columns are the white matter. Internally, in that butterfly H-shaped gray matter area, we have the cell bodies. We have some synapses going on there. All right, and then we have a couple of places where things cross. We have an anterior white commissure. What do you suppose crosses in the white commissure? Hopefully you're catching on to this. That would be the axons. And then we have an anterior gray commissure, again a crossing area. And in there we have cell bodies because it's gray. All right. Now we have one more interesting piece of information here, which we can see. Oh, here we have some circles. All right. Anterior white commissure, anterior gray horn, posterior gray horn. And now we're moving on to something that's a little bit different that only occurs in certain areas of the spinal cord. The little pointy thing on the side that kind of looks like a little horn is called the lateral gray horn, also called the intermediate gray horn. Just to make it more confusing, has that different name, couple of different names. This is special and different. It only occurs on levels T1 to L2. Those are the only ones that have a lateral horn. So when you see that lateral horn, you know that you're somewhere between T1 and L2, levels of the spinal cord. All right, and we'll talk about that as we go on too. So what happens there? This is where we have the sympathetic nervous system. Lying next to the spinal cord, basically along the bones of the spine, is the sympathetic chain ganglion, and this is where the cell bodies of the sympathetic nervous system live. And parallel to that, in that particular area, we have this lateral gray horn where we have, or this intermediate gray horn, where we know that that's pointing us to where this sympathetic nervous system lies. And we'll talk about that more as we go along as well. All right, we have a couple of other anatomical landmarks here. We have a posterior median sulcus. Let's see if that'll show up here. Oh, let's go with lateral and anterior median fissure, since that's what's going to pop up first. So the anterior median fissure is pretty big. Lateral fissure is just kind of more like a little divot, a little bump there. And then we have a posterior median fissure too, not as big as the anterior. All right, and then we have these intermediate fissures as well. So these give us sort of a schematic of what we're looking at here. So why would the anterior one be bigger? It's because there's an artery. The spinal artery actually runs, the anterior spinal artery runs down the length of the column of the spinal cord in that fissure. And that makes that bigger dent. So it actually is a landmark, lets you see to know whether you're looking at the front of the cord or the back of the cord. Now, this is where, as we've already pointed out, or maybe we haven't pointed it out yet, but anterior is motor, posterior is sensory. AM, anterior motor. PS, posterior sensory. So AMPS, AMPS, anterior motor, posterior sensory. And that's all the way through the central nervous system. If you're trying to figure out, I'm in the front, is it anterior, and we're anterior, is that motor or is it sensory? Anterior motor, posterior sensory. Let that help you remember it. Okay, there's the posterior lateral fissure. So a little bit different grooves in the back there. All right. So here we have another look at we're taking another look at the posterior median fissure and the posterior lateral fissure. The lateral fissure is where the nerve roots from the posterior sensory nerve root enter the spinal cord, and it makes more of a groove than what you see in the front. So that groove is just given an anatomical name there. The intermediate fissures along the side are where the posterior spinal arteries run. So instead of one main one, like we have in the front, we actually have two in the back. So we have two posterior spinal arteries, and they're not as big. The anterior spinal artery actually is larger and perfuses more of the spinal cord, more like 3/4 of the area, and those posterior spinal arteries only do about 1/4, so they're smaller. Okay, so let's take a look at how the spinal cord is situated in the bone. So take a look at the kind of on the what looks like it's at the top of your screen, and it almost looks like a pointy hat. That would be the pointy bone, and that's dorsal or posterior. What is that bone called? All right, pulling you way back from anatomy, it is the vertebral lamina. So there's a round bone in the front. Okay, what is that round bone in front of the spinal cord called? That's the vertebral body. So when we talk about discs in the spinal cord, where do you suppose those are? Those are between the vertebral bodies. So they're sitting between those round bones all stacked up, and they're in front of the spinal cord. But notice there's lots of bone all the way around the spinal cord in order to protect it. So let's take a look here at the nerve roots. So what do you suppose that one is? Oh, we got the name of the dorsal root first. All right, so we had dorsal root. That dorsal root is sensory. Posterior, sensory. So these are afferent fibers going at the spinal cord, eventually toward the brain as well. These are separate from the ventral root, which is close to the spinal cord. I got to get my light to come back on. There we go. All right. The sensory information is coming from the periphery, and it has the cell bodies in the dorsal root ganglion. So you'll see this little bump in the back that you don't have in the front. This is the dorsal root ganglion. Sensory nerves have a little bit different structure, and they actually have a peripheral axon that comes to the cell body. There's no synapse in the dorsal root ganglion, and then they have a proximal axon that heads off in the dorsal root going toward the spinal cord. We're going to take a closer look at those nerves later on. Right now, I want you to just get used to the gross structure of where the dorsal root ganglion is, where the dorsal root is, and that they are sensory, and that there are cell bodies in that dorsal root ganglion. These send the messages on to the central nervous system, like I said. Now, there's a ventral root. Let's back up. That the arrow, I probably can't see the word there on the bottom. The ventral root, that is information that is exiting. So that is efferent, anterior, motor, exiting in the ventral root. Okay, these are efferent fibers exiting the central nervous system. All right, now we're looking at an actual real spinal cord here. What side are we looking at? Are we looking at the front or the back? And how can you tell? Well, do you see that big artery running down the front? There's one artery, not two. So what does that tell you? That tells you that we are anterior, and we are looking at the anterior spinal artery that's sitting inside of the anterior median fissure. So we're looking at these roots, these nerve roots, and we're looking at some of them on the front and some of them on the back. Which do you suppose the ones on the front are? Those are the ventral nerve roots, given the name, right? And the ones on the back are the dorsal nerve roots. They're cut off here because, it's a picture of the spinal cord, but we're getting oriented to where we are in the spinal cord. All right, so here's another schematic of it, and now we're looking at the sympathetic chain. Now, do you remember what levels we said that the sympathetic chain runs along beside? It runs along T1 to L2. This is called the sympathetic chain ganglia, the sympathetic chain, also the paravertebral ganglia, and the sympathetic trunks. So all kinds of names for the same thing. These contain the cell bodies of the autonomic nervous system, specifically the efferent fibers of the autonomic nervous system. So are we looking anterior or posterior here? Well, we know we're looking at the anterior because we can see the aorta and the subclavian, and we can also see the trachea and the bronchial tubes. So that's how we know we're looking at this from an anterior view. Okay, this is an important slide. This is one you want to bookmark and keep track of. This is showing what's going on at each level entering and exiting the spinal cord. So first of all, where are we in the spinal cord? All right. Notice that we have a sympathetic ganglion. What does that tell you? That you're somewhere between T1 and L2. Now let's start at the cord and we're going to go out from there. Let's look at that dorsal root ganglion. What kind of fibers are these in the dorsal root ganglion? These are posterior dorsal, so they are sensory. What do they have here? They have cell bodies. Okay, then we look at the ventral root, ventral nerve root. This is sensory or motor. It's motor, okay? And this includes both the visceral or autonomic and the somatic. These, the visceral fibers are going to go to the sympathetic ganglion, where they're going to synapse, and then they're going to head to the areas they control. They're going to go to the visceral organs, which that branch goes down the chain postganglionic. Okay, it's going to go down that sympathetic chain. If it goes to the back, it's going to go to the dorsal ramus, the body wall, and the limbs goes to the ventral ramus. All right, so let me say that again. From the autonomic nervous system, you're going to have things going three places. You're going to have some of them going to the visceral organs, which those head down the sympathetic chain. If it's going to the back, like your back, it's going to go to the dorsal ramus. But if it's going to go to the body wall and the limbs, it's going to go to the ventral ramus. All right, so now let's talk about the somatic motor. These aren't going to go to the sympathetic chain ganglion because they're not sympathetic fibers. All right, they're going to travel through the dorsal ramus. The ones that travel through the dorsal ramus are going to go to the back, and the ones that travel through the ventral ramus are going to go to the skeletal muscles and the body. Okay, so the somatic fibers, it's the somatic motor. You'll see that in the red there. Those are going to be, it's hard because I can't point. Usually I'm like, oh, look here on the screen, and so that's going to be difficult. So if we need to talk through this again when I'm with you all, that's fine too. But those somatic motor fibers are going to, there are two kinds. They're going to go two different ways. Some to the skeletal muscles of the back, some to the skeletal muscles of the body wall and limbs. Okay. Now, notice this, there's this tiny little part between the dorsal ramus, the dorsal root, the ventral root, and the dorsal ramus and the ventral ramus. That's the spinal nerve. The spinal nerve itself is very short, okay? It isn't until after we get past the sympathetic ganglion and get to the ventral and dorsal ramus that they're going to become peripheral nerves, okay? So all this is going on, the splitting and rejoining, kind of combining. It's like a highway. It's like this major highway, and we've got all these intersections and flyaway ramps and things like that. And we're just getting oriented to them now. All right. There's the autonomic sympathetic ganglion. There are two rami communicantes, the white ramus and the gray ramus. The white ramus carries the fibers as they go into the ganglion, or pregangliotic. The gray ramus carries the fibers as they leave the ganglion, or postganglionic. All right. So kind of back to seeing where we were in the whole structure. There's the ventral ramus is the biggest nerve, and the dorsal ramus, which is the smaller nerve. And that's when we're becoming peripheral nerves right there. All right, now we're looking at the whole spinal cord. We got that here. It's what the whole spinal cord looks like. we have the spinal cord starting at C1. And notice this is just continuous with the medulla. The only difference that happens is that the medulla comes to the foramen magnum and it comes out, and when it comes out, it's the spinal cord. So now we have C1 through C7, which are the bones, okay? Then we have the spinal cord floating, and notice the spinal cord is actually shorter than the bones. That's because of how development occurs. I'll talk about that more when we get to the week on development. But the spinal cord actually floats and ends at L1 to 2. There is a cervical enlargement and a lumbar enlargement. Why do you suppose those are enlarged? Well, that's because we have upper and lower extremities, and they take more nerves than do the trunk, so that we can have our fine motor and so that we can have our stepping patterns. So you have these enlargements where there's more nerves in those areas. Okay, the spinal cord ends in the conus medullaris. It has this cone-shaped structure. Coming off of that conus medullaris, in order to have the nerves exit at the spinal cord level that corresponds to, or at the vertebral level that corresponds to the spinal cord, you have this cauda equina, which means it's the tail of the horse. Kind of looks like a horse's tail with these hairs hanging down, and then the fibers exit. Okay, we're going to come back to those. At the end of the conus medullaris, there's a long fiber that tethers the spinal cord to protect it to the coccyx, and it is called the filum terminal. Okay, filum terminal anchors the spinal cord to the coccyx to keep it from getting away, from floating up where it doesn't belong. All right, so how many of each segments do we have? Bones and nerves. Well, with cervical, we have 8 nerves but 7 bones. That's because there are nerves coming out at the top and the bottom of each of the vertebral levels. So we have 8 cervical levels. We have 12 thoracic levels and 12 bones. We have 5 lumbar levels and 5 bones. We have 5 sacral levels and 5 fused bones in the sacrum. And in the coccygeal area, we have 1 nerve and 4 fused bones. Okay? Now the cauda equina, let's come back to that. Those are the nerve roots that have to elongate to reach their points of exit at the vertebral foramina that corresponds to the spinal level. So they're quite long. In fact, they're several inches long. So if you ever look at an actual dissected spinal cord, it's got this tail hanging on it that kind of looks like, a horse's tail, kind of a hairy tail, and that's the cauda equina. All right, so what's protecting the spinal cord? Let's talk about that a little bit. There's some layers to it. Just like we have in the brain, we have the dura mater, which literally means tough mother, okay? And then we have the arachnoid layer, which looks like a spider web. That's where it gets its name. And then the pia mater, the little mother. These are the same levels as are in the brain. We also have cerebrospinal fluid in this area. So if you look in chapters 13 of our book, pages 296 to 297, you'll see this figure. We also have these dentate ligaments. See the arrow there pointing to these little tooth-shaped ligaments. That's where they get their name, the dentate ligaments. Here we have, back to the spinal cord, looking at the sections of the spinal cord. We are below T6, okay, and the posterior column is called the fasciculus gracilis at this point, and it's one big column at this point. Above T6 at this point, we end up getting 2 columns, and we'll talk about that a little bit as we go along. We're going to try to stick with, keeping the information where we need to right now. But just know that below T6, we have this posterior column, and it's called the fasciculus gracilis below T6. And of course, this section is L1. Next week, we're going to talk about what happens above T6. I wanted to show you that this is the sacrum, or S1. What is dominating here, you think? White or gray matter? Well, it kind of looks like we have more gray matter, doesn't it? Why do you think that is? Why do we have more gray matter here than we do white matter? Well, that's because at this point, if you think of the fibers that are going to the spinal cord at this point, a lot of them are above that level. So we don't have as many fibers heading into the spinal cord. We don't have as many highways traveling north, traveling up that spinal column at this point. So there's more gray matter than there is white matter. All right, this would be a good point to take a little break if you want to. The next few slides that we're going to do, we're going to move on up and go on into the medulla and the pons, and that is where we are heading next. Okay, that's L3. Sorry, we have a few more levels here just to take a look at. You can see some of the different layers in the spinal cord, or the levels in the spinal cord. Oh, okay. I had on this slide we have T at the level 2-2. Remember how we said something happens different at above T6? This is actually showing that for us. So now we have on the outside of the black area that's outlined is the fasciculus gracilis. The inside area is called the fasciculus cuneatus. I'll let you think about why does that happen at T6 or above T6. And we'll talk about that more next week when we are together as well. Fasciculus gracilis there in red. And now up in the cervical level, notice how much more white matter we have, and you can see the fasciculus gracilis and fasciculus cuneatus outline. Remember, cuneatus is more medial, gracilis is more lateral. Here we go, moving on up the brainstem. All right, so what angle are we looking at from here? Which side of the brain are we looking at? Let's get ourselves oriented. So if this poor person is lying on their back on a table and we're standing at their feet and looking at the underside of their head, this is what we would see. So their eyeballs and the front of their face would be at the top of our screen, and the back of their head resting on the table is on the bottom of the screen. One of the ways that we can orient ourselves in that way is look for the cerebellum. That's the very striped looking round objects that are sitting down there at the bottom. Another way you can look at it is you can actually see that the olfactory nerves going up in the middle of your screen at the midline, and then you see this X-shaped structure. That's the optic chiasm. Okay, and it's a little more toward the top there. So the top is ventral, the bottom is dorsal. We can see several structures here. Can you see the medulla? That's that cut off structure at the bottom. So if it were not cut off, not dissected, the spinal cord would be coming at us out of that medulla. All right. Then we have the pons, which is the belly shape. Looks like a great big round belly. You always know your anterior when you see the pons because it's a belly. Okay, it looks like a great big belly. Like I said, optic chiasm looks like an X. And then we can see also the underside of the cerebrum and several of the different lobes of the cerebrum. The cerebellum being those ball-like structures at the bottom. All right. Just some squares to show that. Let's get a little bit oriented to the ventral or the anterior side of the brainstem. How do we know that we're on the anterior side? Because we can see the big belly of the pons. That's how we know that we are ventral. Okay? So you've got that big belly. If you don't see that big belly, you're not on the ventral side. So if you're like not sure which structure you're looking at, look for that pons. Now, pons starts with P. Pyramids also starts with P. So those bumps that are below the pons are the pyramids, and they kind of look like pyramids. They're kind of diamond shaped, and that's what gives us their name. These are on page 358 in the text, figure 15.1. Also, if you look at figure 1.9 on page 9, figure 1.10 on page 10, all right, the pyramids are on the medulla. We're looking at the medulla here. It's the part below the pons and above the spinal cord. All right, there's another structure, okay, that is the pyramidal decussation right there. structure I was going for was actually the olive. That's why I got thrown there for just a minute. The olive sits lateral to the pyramids, and it kind of is a bump that looks like there's an olive underneath of the skin there. So our anatomists were not very creative, and they just called it what it looked like. The pyramidal decussation is where some things cross, and we'll talk about that more as we go along. Since we're anterior, would you expect it to be motor information that's crossing or sensory information that's crossing? So remember our amps: anterior motor, posterior sensory. So we have some motor information crossing here. We'll come back to that later. This here is a cross section of the medulla. All right. In this cross section of the medulla, what kind of a section are we looking at? It's called transverse because it's as if you were cutting this way. Okay, it actually is probably more like up about this level. All right. Which side is anterior or ventral and which side is posterior? Well, we know that the pyramids are anterior, right? Pyramids, pons, starts with P, anterior, anterior motor, okay? So what kind of information do you think is going through those pyramids? If you're guessing motor information, you are correct. All right. Pyramids in a cross section or transverse section look like they're kind of rolled over on themselves or folded over on themselves because that's actually how they have developed. That's actually how they look. Okay, moving on. So this is an in situ view of the brain, or obviously this person is no longer alive, but we're looking at an actual brain. What do you suppose all those fiber-looking things coming off of the medulla and the pons are? Those are cranial nerves. So if you look at figure 14.1 on page 329, you'll see the cranial nerves. Now, can you find cranial nerves 2 through 12? They're actually all visible here. So take a little time. You can maybe pause the video and see if you can find them. Use table 14.1 on 330 to help you get the names right, and the figure 14.1 on page 329 to help you with that. You also can see them in the atlas on pages 480 and 481, and that will help you with that. All right, so what are we looking at here? If we're looking at the pyramids, we are looking at another cross section of the medulla. All right, so we see the pyramids. The medulla oblongata is its full name. We know we're further up in the medulla because the pyramids are larger. So the pyramids are how you recognize this cross section as the medulla easily. We've got some white matter spots going on, but we see those whitish spots. They're not necessarily white matter. We'll talk about that as we go on too. But you can see the pyramids as those kind of triangle-shaped structures at the bottom that let you know where you are. All right, so we've been looking at the anterior view of the medulla and the pons a little bit too. Let's look at the posterior view. We're going to look at the dorsal side of the brainstem. Now, instead of 2 mountainous structures that are the pyramids in the front, we have 4. These guys are called tubercles, and we have 2 on one side, 2 on the other. So in the center, we have the gracile tubercle and the cuneate tubercle. Oh dear, I think I told you backwards before. Oh dear, because I was telling you in, let's go back. I am so sorry. I don't think I want to re-record the whole thing though. All right, in here, the black I told you was cuneate or fasciculus cuneatus. That's fasciculus gracilis. It stays medial. I totally messed that up, and I'm so sorry. So, fasciculus gracilis medial, fasciculus cuneatus lateral. I'm human. I guess I'm going to make a few mistakes. Let's go on down here. Okay, because that corresponds to the fasciculus gracilis corresponds to the gracile tubercle, because it's just a continuation of the spinal cord, and the fasciculus cuneatus corresponds to the cuneate tubercle. Okay, I promise we'll get this straight as we go along. All right. There's no belly of the pons here, so we know we're posterior. We see some other kind of strange structures that we'll talk through just a little bit. We have these peduncles. Now these are these sliced off structures that almost look like you took someone has a hair braid and you took and cut it off, and now you've got this cut off spot. Those are called the peduncles, where they're sliced off for us to be able to see the backside of the brainstem. They're actually attached by loads of fibers. What do you think they're attached to? Well, they're attached to the cerebellum because that's normally what's sitting back here. All right. And I think that's all I want to say from this view. There's the arrow, the inferior cerebellar peduncle. So there, even though it looks like there's only one round structure there are actually three: inferior, superior, and lateral. But that was just showing you the inferior cerebellar peduncle, because that's actually what's in the medulla. Okay, inferior cerebellar peduncle, that's what's in the medulla. Okay, so now, where are we? Still in the medulla. What kind of a section? Transverse. How do we know we're in the medulla? Because we have the pyramids. Okay, but we can also see the other two structures that we've talked about, the gracile tubercle and the cuneate tubercle, that corresponds to the fasciculus gracilis, which is medial, Brenda, it's medial, and the fasciculus cuneatus, which is lateral, found above T6 in the spinal cord. Okay. And here's another view of them. Gracile tubercle, cuneate tubercle. Now, are we dorsal or ventral? We're dorsal. How do we know? Because we don't see the pons, and we do see the peduncles. Okay, we also see another structure that's going to be really helpful to us, and that's this kind of kite-shaped or triangle-shaped or more diamond-shaped structure here. That's the fourth ventricle, and we're going to talk about that as we go along a little bit too. Now, here we have two views put together in the same schematic. On the right is the dorsal view, and that shows what happens when the peduncles are not cut off. So on the left side, it's cut off, and on the right side, you can see how the cerebellum is attached. And those peduncles are sending fibers to and from the cerebellum to help us with our balance. So we can kind of see how it's situated a little bit, and we can also see how it's situated from the side. What's a side view called? That's a sagittal view. Since this is right down the center of our brain, what kind of a view is it called? Mid-sagittal. Now, another landmark that we're looking at in the medulla oblongata is the fourth ventricle, like I said, that kite or diamond-shaped structure. And this is surrounded by the inferior peduncles, and it lets us know we're dorsal. And that's the fourth ventricle. Now, you can also see the fourth ventricle in the sagittal view, and it kind of looks like this pointy structure heading into the cerebellum. It's this kind of empty space heading into the cerebellum. So, there is one other thing I want to point out here in the sagittal view is this dark line running down the spinal cord. What do you think that is? That is the central canal. And that is for the flow of cerebrospinal fluid. You can actually see it in cross sections of the medulla. So let's go up a couple here. You see that little hole right there? That's the central canal, right there in the center. And that is where cerebrospinal fluid is flowing from the fourth ventricle down to the spinal cord. The fourth ventricle has 3 outlets, 2 foramina of Luschka. Let me see if I can get an arrow. Nope. Okay, we'll stay here. 2 foramina of Luschka, which are lateral, and 1 foramen megendi, which is medial. Now, these are draining into cisterns that we're not going to talk about just yet, but just so that you know that there's some flow going on there, and there are some holes going out of that area. Okay? Foramina of Luschka, the 2 lateral, and 1 foramen megendi, medial. Okay? And then we have the central canal heading down the spinal cord. These are all containing cerebrospinal fluid, which what does cerebrospinal fluid do? It's our shock absorber, and it's also has a cleaning function for our brain. Gets our brain cleaned up, We're going to learn more about that later. And here's just another view of the fourth ventricle. It's showing all the ventricles. The one that's way down low, kind of between the cerebellum and the medulla and pons, is that fourth ventricle. And that's where that cerebrospinal fluid is at. All right, we're going to be looking at that more in 2 weeks, but I just kind of wanted to get you oriented to seeing that kite-shaped structure between the medulla and the pons on the dorsal side. All right, let's look here at some cranial nerves. Now, this looks a lot cleaner than in our actual brain pictures. In the medulla, we can see 9 through 12. So we've got cranial nerve 9, which is the glossopharyngeal, 10, which is the vagus, 11, which is spinal accessory, and then more medial, we have 12, which is hypoglossal. Those are the 4 cranial nerves that you can find in the medulla, and you should be able to recognize those on sight. Hopefully you're already a little familiar with them, and this is just a review. So now we're going to go onward to the pons and get in a little bit about the pons. All right. We have, we're going to look at a couple more cranial nerves here. Glossopharyngeal nerve again, vagus nerve, 9, 10, 11 accessory, 12 the hypoglossal. So that's what it looks like in real life, on a real brain. Okay. Now, back to the pons. So we actually have this groove in the front of the pons that is called the basilar sulcus. And that just shows you where the midline is on the pons. So now we're looking at the dorsolateral pons, and we have a few structures here. How do we know we're dorsal? Once again, we have the peduncles. Now the one that is associated with the pons is the middle cerebellar peduncle. Notice there's not really a fine defining line between the inferior one that we looked at before and the middle, but it's just kind of pointing to the middle, and it's the middle cerebellar peduncle. Also the brachium pontus, but I don't usually call it that. You'll find me calling it the middle cerebellar peduncle. All right, it's in the same round part as the inferior peduncle, but it has a different name because it's anatomically associated with the pons. There's also a superior. Where do you suppose that comes from? That's going to be the midbrain. So we'll get to that midbrain later on. So this is the dorsolateral pons. Now we're back to the anterior pons. Let's talk about the cranial nerves coming off the pons. So we have 5 through 8. 5 is going to be the trigeminal, and it's a pretty big one because trigeminal means it has 3 parts. 6 is the abducens. 7 is the facial. And 8, a favorite of mine, the vestibulocochlear nerve. All right. So we have all the cranial nerves so far that we've looked at have all been on the ventral side. Notice we can see 5 through 8, also 9, 10, 11, 12. You remember what they're called? You could quiz yourself now. Finally, we are at the end. Here's what the cranial nerves look like on an actual brain. Can you find 5, 6, 7, and 8 by looking at it here? Okay. 5, 6, 7, and 8. Okay. Next week, we're going to look at the midbrain, the basal ganglia, and the cerebrum. All right. Don't forget to do your reading review assignment and do that before class next week. Oh, here we go. They're labeled. Awesome. Okay. And there's our references. Thank you for joining me in this way. Sorry, it's kind of an awkward format, but we will